Hello friends, so how you doing? And I hope you really are having a good time, after the last session. So today friends, this is Dr. Ashmir and today we will discuss, the CBSE 2018, some of the questions in the sample paper which might be of, of much utility to you, right? They always say that, practice makes a man perfect. So, after doing the theory, let us revise and let us do some of the questions which might come for your CBSE exam. Okay, so let us continue. The section A consists of basically 5 questions which will be mainly of 1 mark. You have to be to the point here and you have to write exactly what it means. But here we will talk a little in detail, I'll explain you in detail and I'll write the main things which need to be written over here. So friends, here is your first question for the day. How many chromosomes? How about if it would have been like the KBC? Here comes your first question. How many chromosomes are in a gamete of an organism that has 46 chromosomes in its cell? Before you do this, before you read this, we all know that in an organism, okay, I'm just explaining you right now. It has nothing to do with the writing. Okay, you should not write so much in one marker, only to the point. So the answer, I'll write it as in an organism, okay, in an organism the cells will be having the 2n number, will be having, okay, in an organism the cell will be having 2n chromosomes. Right? Now, for example, now each of the cell will be having 2n chromosome. Now what will happen when the gamete formation is taking place, my dear friends? These chromosome, what happens? So what will happen? There will be meiosis of the gamete. There will be meiosis and the gametes will be formed. The gametes will be formed. Okay, so because of the meiosis the gametes are getting formed and hence these gametes will contain this, will hence contain n number of chromosomes. Okay, this will hence contain n number of chromosomes. Got it? So in an organism the cells will be having 2n number of chromosomes. What will happen? This will undergo meiosis when the gametes will be formed and hence the gametes will have only n number of chromosomes. So now we will apply this logic and we will see here that how many chromosomes are in a gamete of an organism that has 46 chromosomes in its cell. Okay, so now I will write this as 46 chromosome in cells, okay, 46 chromosome in cells, gametic meiosis, right, and hence in the gamete there will be 23 chromosomes formed. Okay, hence in the gamete 23 chromosomes will be there. Now what is the why 23 chromosomes are formed? Okay, let us understand this. My dear friends, when the fertilization will take place, we all know that when the sexual it is talking about the gamete. So this type of reproduction is definitely sexual reproduction. Why? because when there will be a gamete, male and female gametes will be there. These male and female gametes will fuse. So say for example, an organism is there and it has a body in an organism. There is a body, the body of the organism has 46 chromosomes, okay, 46 chromosome. Now if that organism will produce a new organism, that should also have 46 chromosome. So, and it is the sexual reproduction is taking place. So the male and female gamete are combining, the male gamete plus the female gamete are combining. So if this 46 chromosomes do not undergo, the half, do not get reduced to half, then what will happen? The male gamete will also have 46, the female gamete will also have 46, and the resultant will be, the new organism, the offspring will have 92 chromosomes, and this way it will keep on increasing. That becomes null and void, that holds no significance. So that is why we say, okay, the male gamete, when the gamete are formed, this should get reduced to 23, this should reduce to 23, and this also this also should get reduced to 23, and hence the whole thing should now become what? 46, right? So I hope you understood. Okay, this is a very simple concept. Okay, so you will write your answer just in a word form. One word you should write, one sentence you can write, that the chromosomes in a gamete of an organism that has 46 chromosome in its cell will be 23 chromosomes formed in the gamete. You can write it this way, or you can form a sentence, that would be better, right? So I hope this is clear. And now we move on to the next question. So the next question for rupees 20,000 is here on the board. It is often seen, it is just for 1 mark, okay, don't get carried away. It is often seen that replication of DNA does not initiate randomly anywhere in the DNA, right? So it will not initiate anywhere, it instead starts a definite region. Why is it so? And give the reason. So see, the replication of the DNA always starts at the region called the origin of replication, ori. Okay, so I'll write this here as the replication of DNA. Okay, the replication of DNA starts at a specific site. Okay, starts at a specific site. Okay, it starts as a specific site called ori. Now what is this ori? This ori is nothing but the origin of replication. Okay, this ori is nothing but origin of replication, called as the origin of replication. That means what? The replication is starting at this point of the DNA. Clear? And now why is it starting here? Because it is at this point. Okay, it starts at this point. It starts at this point. At this point, because a protein complex, okay, a protein complex called pre-initiation complex, okay, called pre-initiation complex, will bind here, and what does it do? Will bind here, and this will recognize okay, and this will recognize, okay, and this will recognize the the unwinding and replication of DNA. This will recognize and start, okay. Got it? So once again, the DNA, okay, just look here, I'm just drawing it. The DNA, okay, this is not the right diagram. We all, how is a DNA, right? It is helically coiled, double-stranded, it's coiled like this. So it has to be uncoiled. Only when it is uncoiled, these two coils, when they get uncoiled, right? These two, when they get uncoiled, it will look like this. One will act as a template strand, and on this template strand, the other one, that is a daughter strand, will be manufactured. So we see here that the uncoiling has to first take place for the replication of DNA. So how does it happen? So we see here that the DNA replication will start at only a certain point of the DNA. That point is called the origin of replication, the ori. And why it is happening at the ori? Because at ori there is a protein complex called the pre-initiation complex, which will identify and start the unwinding and the replication of the DNA. Got it? 1 mark, more than enough. You have already won 20,000, and let us now go into the next question. So the next question for rupees 50,000 is here for you. Give one adaptive feature found in the Amniocentesis flowers. Are you ready in your hot seat? Do you want a 50-50? No, you are confident enough, and so am I. So the answer would be Amniocentesis flowers, first of all, are flowers, which are what? Wind pollinated flowers. Wind pollinated flowers, right? That means the wind is playing a very major role in the pollination of these flowers. And what is pollination? Pollination is the transfer of pollen grain from the anther to the stigma of a flower, right? Now, since the pollen grain here has to be taken by wind, because these are wind pollinated flowers, so these, these pollen grain, what are the features? The pollen grain over here, the pollen grain in these flowers should be produced in large numbers, okay? Should be produced in large numbers, right? It should be produced in large numbers. It should be dry. They should be small. Okay, it can be dusty. Okay, dry, small, dusty. Why? And it can also have some feathers or wings, because they can be carried away by the wind, right? So these should be produced in large numbers. Why produce in large numbers? Because see, these are carried away by wind. You never know where these will fall. You say out of the 100 pollen grains which is being carried away, 5 or 10 may only reach the flower, and the other 95 may go for a waste. To you may never know, all 100 may go for a waste, or all 100 may go on the flower. So to increase the chances, they have to be produced in large numbers. They have to be dry so that the weight is less. They have to be small, and dusty so that they can be easily carried away by the wind. Amniocentesis flowers. Got it? So you have 50,000 in your kit. Do you want to leave the play right now? I suppose no. And so we move on to the next question. What is amniocentesis? What is amniocentesis? Phone a friend. I don't think so, because you know this and this is just a one marker. This will give you 1 lakh rupees in your account. Okay, I'm going to give you the check, don't worry. You can always contact me for the checks. Okay, amniocentesis. Amnion is covering, the embryo in amniotes. Amniote is a special class of animals, okay, where the embryo is surrounded by amnion. Okay, so I'll write this here as the covering around the embryo is called, the covering around the embryo is called as amnion, right, which contains a fluid called amniotic fluid, okay, called amniotic fluid, right friends? So the covering around the embryo is called as the amnion, and which contains a fluid called the amniotic fluid. Now this amniotic fluid, what they do, they take out this fluid in the process of amniocentesis. This is a fetal test done in the fetus. Okay, what they do, they take out this amniotic fluid and they detect, they check for the chromosome, for the abnormality in the chromosome. And recently it has been used, not recently, since a very long time they have been using this for the detection of sex, that is the sex of the fetus, which is absolutely wrong. Okay, so I'll write this as amniocentesis is a fetal test, that means a test done in the fetus where the amniotic fluid is being tested, for the chromosome, to know the sex of the fetus, and other abnormalities. Got it? So amniocentesis is a fetal test which is done in the fetus, and where the amniotic fluid is taken out with the needle, okay, and they check for the chromosome, because they will have the chromosome, the amniotic fluid will have the chromosome, to know the sex of the fetus and other abnormalities. Knowing the sex of the fetus by the amniocentesis is a banned procedure. You cannot do that. You cannot check the sex of the fetus, because both the male and the female have the right to live in this world. But you also have the right to know this question, because you are the owner of 1 lakh rupees now. Okay, we move on. What do you think the next question will be? This is the master question of, section A, which is, so the next question appearing on the screen will be of 1 lakh rupees, and the question here is, list 2 advantages of inbreeding in animals. Okay, list 2 advantages of inbreeding in animals. Inbreeding, that is breeding in the same, in the same line, breeding in the same dynasty, same class. You're not breeding with something else. So the 2 advantages of breeding will be, the 2 advantages of inbreeding will be what? In animals will be, the first one, it will produce a pure line. It will produce a pure line of animals, a pure line of animals. Okay, it will produce a pure line of animals. What do you mean by pure line? For example, if I have a basmati sona rice, okay, let me write both of them, then I'll explain you. The second one, it will produce homozygous animals. It will produce homozygous animals, homozygous organisms rather than, because it can be done both for plants and animals. Okay, homozygous. Now, what do you mean by pure line of animals? See, for example, if I, we all love basmati rice, right? If I say that, I want more of basmati rice, I'll do what, I'll take a strain of basmati rice, I'll breed it, okay, all with the same, the same strain of basmati rice, and so over a period of time, what will happen, since the crossing or the mating has been only with 2 strains of basmati rice, over a period of time, say 2, 3, 4 years, whatever, the whole plantation will have basmati rice. And that we mean by pure line. We are not breeding outside, we are breeding within, and hence, the whole of cultivation will be having the same line of organisms. Okay, this should be organisms and not animals. Right? So, a pure line. And homozygous? Slowly and gradually, if say suppose someone is AA and BB, over a period of time what will happen, the whole of the organisms will have the same genotype. Okay, it will start having the same genotype. Why? Because there is inbreeding, we are not breeding from outside. Now what would be, this is enough for a 1 mark answer. What would be the disadvantage of this? The disadvantage of this would be, say for example, a disease comes, or there is some calamity, whatever, an infection, a viral infection. So that viral infection, because all of these, these pure line will be having the same genes, the same organisms, the same genes. So say suppose, there is no variation here, boss, and if there is no variation, any virus or infection comes and it will affect, say it affects this gene, then the whole cultivation of basmati will wipe out. Then how will you eat basmati? Okay, so this is one of the disadvantage of the inbreeding. I think you are already a winner of 1 lakh, right? Do not get excited, there is much more to win. So we will probably stop here with section A, and then we will continue with section B, right? So we are going to continue now with section B. I hope you enjoyed this session, and hope it was a very enjoyable one. Hope to meet you again with Dr. Ashmir. A very goodbye from here. Thank you.